Today I'm going to give you an introduction about CRISPR toolkit development for natural products discovery in actinomycetes. And a little bit background of myself. So now I am in Denmark. Just as introduction, I'm in Technical University of Denmark. We are in this very international institute, Novo Nordisk Foundation Center for Biosustainability. It is located in Copenhagen area. And this is the nice building I'm in. And the section I come from is called New Bioactive Compounds section. So it is headed by Professor Sandhya and Professor Tim Weber. So what we are doing is we want to combine the power of biotechnology and informatic technology. So it's biotech, BT, and IT to try to find more antibiotics against primary gram-negative pathogens. And we have a nice setup flow from computational biology to wet lab. So the background is that, actually the drug shortage, as well as drug-resistant pathogen is a huge problem for our human beings. And as the statistics saying that actually every year the mortality caused by drug-resistant pathogen is more than cancer do. And the situation will get worse and worse because we simply don't have enough effective drugs, antibiotics. So that's why many people are saying that we need to find or develop more antibiotics for this kind of purpose. Saying is superbugs need drugs. And the thing is that we know that more than half of the antibiotics and anti-cancer drugs are coming from a bacteria called actinomycetes. So this picture shows very nice morphology of this organism. And during last century, we have a golden age of natural products-based antibiotics discovery. So it's basically from 1950s to 1960s. And suddenly after that period, so the speed and output of natural product-based drug discovery drops very fast. So the reason is that the lower hanging apple or fruit can be picked easier than those higher hanging apples. That's why now, because all those lower hanging apples have been picked, so we need some new tools, higher ladders to pick those higher hanging fruits. And what is those higher ladders? That is some new tools, some new technologies. And as you know that the sequencing becoming cheaper and cheaper. That's why now we have accumulated so many genome sequencing data, and this is stationary increasing. By this kind of nice data we have, now natural products discovery has been moved to bottom-up strategy instead of the classic top-down strategy. So simply saying that bottom-up is that we start from genetic information instead of we just start from the cultivation. And then we use modern technologies to do genome mining, and then based on the genome mining information, we do cluster cloning, then put the cluster into a cell factory, then use modern analytic method to finally get the expressed compound, and the compound can be some drug leads later on. If lucky enough, this kind of leads can be later on become drugs. So based on the genome mining information, we know that actually more than 80% of the gene clusters. So gene cluster means that, like, because these molecules is a very complicated pathway, so there involves multiple genes, and those genes normally are clustered. So we call it gene cluster. For example, one compound synthesize one compound normally needs 10, maybe 20 different genes cooperating, and then those genes clustered as a cluster. And based on the genome mining, we know that most of those clusters has not been assigned with any known compound. That means we have a high chance to have some new compounds out of these clusters. But the question is that how to get them from genomic information to final compounds. And I want to show you that this classic bottom-up way. So start from the strands. Now we have a high quality of genomes assembled, and then we can use some modern genome mining technology to get the genome information analyzed. And then from the genome information analysis, then we can have some cluster be predicted, and then this kind of big data, then we need to use laboratory work to do the evaluation. And now I want to give you some timescales, how these steps will be. For example, the first step from the strand to assembled genome maybe takes weeks to months, depends different like growth speed and the facility of genome sequencing. And then after you get in the genome information, assemble the genome, then you do genome mining, because nowadays the computational power becoming faster and faster. So this kind of analysis maybe takes just seconds, 2 hours, maybe days. And then the last step is that from those predicted cluster information to the real compound, it takes years. And maybe you will never get the compound out. So now we see a big gap in between those computational biology to real laboratory work. There's a very huge gap in between. And because we can use computational biology to accumulate more and more data, so-called big data, but then we need to use our own hand to process this kind of big data. And that is just, we cannot do as faster as computer do nowadays. That's why I want to contribute in this area to develop some good tools, try to speed up this kind of processing. So this is old Chinese saying that if you want to do a job, you first need to have some good tools. And we know that before CRISPR, so doing genetic manipulation of actinomyces is very, very hard. 1 or 2 reasons for this is that actinomyces is a kind of gram-positive bacteria with a linear chromosome, and the genome is very high GC. I mean, very high is around more than maybe 70% of GC content. And if you are doing a molecular biology, you will know that if DNA fragment with 70% high GC content, even the PCR will be a problem, will be problematic. So that's why the classic method is very complicated, and I'm not going to give you the details. I just want you to know that it is time consuming and the efficiency is low, and it's a double crossover based method. And like just kidding, I'm lazy, so I need something new to speed it up. And then when I step in this area, that is the later of 2013, and that is a very good time of CRISPR, and just CRISPR was emerging at that time. And this is how CRISPR works for genome editing. So simply saying is that first we have nucleus, for example, Cas9 or CPF1. So now we call it Cas12a. And then we give it a piece of guard RNA, so sgRNA. So sgRNA and this nucleus, they can form a complex. And this complex will kind of a scanner to scan the genome. And then if it finds a target, so the RNA, the spacer RNA in within this sgRNA will just pair to the target, complementary to the target DNA. Then this will cause the conformational change of the nucleus, and this nucleus will generate a double-strand break of the DNA target. Then after that double-strand break, so there will trigger the double-strand break repair pathway of the organisms. And during the DNA repairing, so we can have different output of genome editing. For example, we can use some defective non-homolog endogenin to have a random size deletion, or we can use non-homolog endogenin, I mean the complete non-homolog endogenin. Then we can just introduce some indels, small deletion, insertion, and substitution. Or we can provide the repair template. So we call it homolog direct repair. So that means we can do it as a cut and paste way. So it is a very precise and high fidelity genome editing. Now it comes to actinomyces, and just like I just explained, so the magic of CRISPR-Cas9 genome editing is inside of the double-strand break repair. And I just mentioned there are two major ways. One is non-homolog endogenin, and that is the dominant way in eukaryotic cell, and the homologs recombination is used by most of the bacteria, because most of the bacteria is the non-homolog endogenin pathway is missing. So that will be a problem. So it's to be or not to be. So if you have a double-strand break, if you don't repair it, it will die. So that means it will force the bacteria to do the repair to survive. And this nice organism is a modern organism of actinomyces called Streptomyces silicola. So this blue compound is actinorhodin. So and we use this to have this proof of concept of CRISPR-Cas9 system. And this is the design of this system. So as you know that in actinomyces, there are not many options of plasmid. There's not many choices. So that's why I put two major components into the same plasmid. And this is temperature sensitive plasmid. So temperature sensitive means that if you increase the incubation temperature of the target strain, so the plasmid, the repair count will be dysfunction and it will be killed, it will lost. That's a very good feature. That means you can repetitively use the same plasmid, the same backbone for multiple manipulations. And this is a shuttle plasmid, so it's E. coli and actinomyces. I mean, because in actinomyces, we normally use the conjugation to use E. coli as a donor to do the plasmid transformation. And this is the major important component, Cas9. And then here is the sgRNA part. So the Cas9 is under control of tetracycline. No, it's a type A, so it's first chapter inducible promoter, and the sgRNA is under control of constitutive promoter, or ME. And just remember that I also have a sort of backdoor for some other components cloning to make the plasmid still all-in-one plasmid system. And just I mentioned that because most of the bacteria, it doesn't have the non-homolog adjoining pathway. But interesting enough, some of the bacteria has, and for example, the actinomyces and the silicola, it has this Ku protein, and it has most of the function proteins of this ligase, but some parts is missing. So this is a LIGD. So there's two major components for non-homolog adjoining pathway of bacteria. So this Ku protein and the LIGD. And the LIGD is a multifunctional protein, and it has at least 3 domains. So in silicola, we can see that Ku is there, and the primase is there, and there are so many DNA ligase floating somewhere, and some other parts are missing. So we saw that probably we can take advantage of this not perfect non-homolog adjoining pathway. So we just gave it a try without providing repair template. That means the cell only can repair the double strand break by its internal mechanism, not by the repair templating. So here's the primary result. We can see that actually, if we don't provide homologous recombination template, the CRISPR-Cas9 worked. So here I want to explain you a little bit. So as I said, that actinirodin is blue, and in this color, so it can produce 2 types of pigment. One is blue compound, actinirodin, and the other compound is red one, and the red one is not diffusible. That means if both red and blue are present, only the blue color can be seen, and the blue is diffusible. You can see that it can diffuse to the media. So you can see here, these 2 clones, it shows red. That means the blue compound, the target cluster has been inactivated. And also we can see from the spectrum, and it is actually, the blue compound is gone. And the blue compound, the pathway was the target that I use CRISPR-Cas9 to attack. And good, and we had some statistics of the efficiency. So the efficiency is pretty much varies from 3% to around 50%, because of different sgRNA gives different efficiency. That is common knowledge. And honestly, this efficiency is not good enough, only 50%. And next step is that we want to see why this efficiency is is not as we expected. And then we just randomly pick some clones for whole genome sequencing to see what's going on with the editing, if that editing is precisely introduced or just some side effect caused somewhere else. So we whole genome sequenced the 4 randomly picked clone, and funny thing is that we found just from these 4 different clones, we saw 4 different patterns. One is just, you see only one single base insertion in this cluster, and this one base pair insertion can just frame shift the whole cluster, and that's also no blue compound can be produced. And in this extreme case is that around 37,000 base pair has been deleted. And you know that the actinium cluster is only 25,000 base pair. And that means the whole cluster has been deleted and beyond that. So we were so exciting by the time to see this kind of unusual result, because at the very beginning, we know that this non-homolog endogenesis pathway is not complete. And that means this kind of becoming a part of competition. So when you have a double-strand break, so the cell want to survive, so it want to repair it. But in the meantime, there were many of the exonucleus in the cell, and they want to digest the chromosome because you have a double-strand break. And by that time, so it's kind of competition. So that's why like it will give you a random sized deletion. So theoretically, it can be in between of 2 essential genes, because if you destroy the essential gene, the cell will die. Even if you repair it, it still will be dead. So that will be a very good application, that because you just need one set of experiments, and if you picked enough clones, you can get a library of random sized deletion, and you can use this for different purposes. For example, to study the gene function, essential genes, and cluster identification, and boundary cluster boundary identification, and so on and so on. So the next step is just I mentioned, because this non-homolog endogenesis pathway is not complete, and we know that which part is defective. So we just want to repair this non-homolog endogenesis pathway, and luckily we by blasting we found 4 functional liquid homologs gene somewhere, and then we just synthesize it and put this cassette into the same plasmid. Just I mentioned that there's rejection enzyme sites that I can use for further things cloning. So still, this is one plasmid system, and I put this liquid expression cassette here, and to complement the non-homolog endogenesis pathway, and then now we see a much better output. So after randomly picked 8 different clones, you see most of them is just a small insertion or deletion, so it's in there, just like a mammalian cell. And then you see that the editing efficiency is also increased a lot. So from 3% to 50%, now we can see around 70–80%. And that's a very good efficiency. And the next step is just I mentioned that there's the other way. Just use the editing template to do a homologous recombination repair, and that is paste and cut and paste repair. So for example, I just provide the left arm and the right arm of the target gene, and after adaptation break, so the cell wants to survive, so it has to repair, and it can use this homologous recombination to repair it, and just to kill this target gene out, and to have seamless deletion. And now from here, just 10 randomly picked clones I can get 10 randomly positive results. And this is around nearly 100% efficiency. And that is also consistent with other reports. So now we have a long story about this active CRISPR-Cas9. So we have different ways, different repair mechanisms. Then we can have different outputs. The next thing is that, you know, in some metabolic engineering, to have completely gene knockout is not a very good way. For instance, if you found later on this gene is not the one you expected, that means you had the wrong knockout, and then you need to start over. But then you cannot use the strain because the strain already has the permanent deletion. So that means that will be problematic. So here comes this knockdown system. That means the genetic information will be still intact. Just the gene expression has been knocking down. And luckily that this CRISPR system also can do this job. So if we just mutate the Cas9, because Cas9 only has 2 active sites for nuclease activity. If we mutate both, this D10 and H400, 840, then it can still bind to DNA, bind to the sgRNA, and the complex still can bind to DNA, but there's no nuclease activity. Then this complex can be served as a roadblock. That means the DNA, the RNA polymerase cannot move if you have this block. That means the mRNA cannot be synthesized. That means the protein cannot be translated. That means that the pathway is sort of inactivated. So this is a nice system. So just as the RNAi, so people call it CRISPR. So it's a CRISPR interference system. And we designed different sgRNA, both from the positive strand and the negative strand, target the same cluster, actin-rodin cluster, the same gene, this ACT1F1. And we saw that just, you know, that if the pathway is inactivated, so the red compound can be seen, so it's red. And because I said that this whole system is based on temperature-sensitive repricum, which means it is reversible. So if you just increase the temperature from 30 to 37, so see the red becoming blue again. So this is a reversible gene expression modulation system. Now I can give you a first summary of what I did for this CRISPR-Cas9 system. So we can just do a very simple comparison to the classic PCR targeting system. So the major advantage is that it is very quick, very fast. If you want to use this CRISPR-Cas9 system for knocking out a gene, it takes 3 weeks. And if you use the PCR targeting, probably it needs 2 months. And also it is highly efficient. Like it's if you use this homologous recombination repair, it will be around nearly 100%. So it is very good. But very soon, when we are using this CRISPR-Cas9 to do some other knocking out in other strains, for example, this Streptomyces caulinaris, we had a problem. The problem is that we tried many times and we just cannot get the expected knockouts. So some background of this cluster. So this is a cluster for synthesized compound called chiromycin. And as I just mentioned that most of the stromyces, it has linear chromosome. And linear chromosome, most of those linear chromosome has inverted repeat at the chromosome end. And this cluster, the chiromycin cluster, located in the end of the chromosome, inside of the inverted repeat, that means it has 2 copies. And 2 copies, we can just think that if we introduce a double strand break in one gene, because this has 2 copies, and it will give you 2 double strand break. So 2 double strand break means that you see a chromosome, linear chromosome with 2 double strand break. That means you have 3 repeats, and we saw that this must be too much for a bacteria to repair it in time precisely. That's why we go back to see after sequencing this, we observed that instead of seamless deletion of KRN gene, the entire cluster was lost. Both of the clusters has been deleted. So that's very strange, and we thought probably that's because of these 2 simultaneously introduced double strand break. So that is the motivation that behind we want to establish something new, some new technologies that has more or less the same efficiency, but will not introduce double strand break. And this is CRISPR base editing. So we named it CRISPR-Best. So it's base editing system. And it has 2 types of base editors. One is from C to T, and the other one is from A to G. So long story short is that the same concept, we just fused the deaminase to CRISPR-Cas9 enzyme, but it's not active enzyme, and it's not also dead Cas9, but it's a Cas9 nickase. Nickase means that it can only introduce a nick in one strand, not both strands. And this is good for recruiting the DNA repair mechanism later on. That's why we use this nickase. So it has editing window, and it can do a lot of things. So this is a summary that what can base editors do. So you know that just from CG to CG pair to TA pair, or AT pair to GC pair, almost all kinds of amino acid exchange can happen. And in particular, that some amino acid can be introduced as a stop codon. And you know that stop codon is very useful. Like there are some possibilities to introduce stop codons. So stop codon means that if you give a stop codon early enough inside of a gene, that means this gene will not properly translated later on. That means the protein will not give you a full functional protein. And that is sort of equal to knockout. And later on, we did some deep characterization of this system. So we want to figure out the editing window, for instance, and which kind of genetic content will be the optimal genetic made-up for base editing. And from the result, we can see that for C to T base editor. So the editing window is around 7 nucleotide. And for the A to G is around 6 nucleotide. And the best genetic content for CRISPR-C-Best is T, the C behind the T. The same with the CRISPR-A-Best is the A behind the T. Then we want to have some proof concept. So showcase one is we still use the model organisms of Silicala. Just I mentioned before that this blue compound, if you inactivated the blue compound, so the red can be seen. And the same, we can see that 4 different sgRNAs targeting this cluster. We see that the stop codon has been successfully introduced. So case 1 and this one is stop codon introduced, and number 2 is here. And we see that some other amino acid exchange happening, and that also will somehow disable this protein. And then this is the other gene inside of this class, also stop codon. And this is also the amino acid exchange. And in Silicala, we saw a very good output of this CRISPR-based system. Then we want to go back to solve our problem of chiromycin cluster, because I just mentioned that use CRISPR-Cas9, we can never get desired in-frame deletion of the KRN gene. And now we used CRISPR-Cas9, CRISPR-based system to introduce stop codon, and we see that it is very clear. First piece of evidence is that we can use PCR to get this fragment out. As you know that use CRISPR-Cas9 because the whole cluster has been lost. That means if you want to use PCR, there's no template. That means you can never get a PCR product. And here, the first piece of information is that I can get the PCR product and can get it perfectly single sequenced. So, and stop codon has been introduced. And the second piece of information is evidence is we use the bioactive guided evaluation, because this KRN gene, if we knock out, we inactivated it, we know that the chiromycin production yield will be much reduced compared to the wild type and the empty plasmid strain. And we know that chiromycin is active against bacillus. So we see the bacillus and we see that yes, the yield is reduced. And then the last step is the last piece of evidence. You just use LCMS, and we see that actually the compound yield is indeed reduced. And then we now know that CRISPR-Best can do some task that CRISPR-Cas9 cannot. So it's good. And then we also had a very nice setup of pipeline for off-target effect evaluation, because you know for CRISPR-related genome editing, the first question or the first concern will be off-target effect, because this is a nucleus and you know DNA nucleus, and it also will maybe cut somewhere else. So this is the pipeline we set up for genome-wide off-target evaluation. So we use the Illumina sequencing to have the genome sequenced, and then we established algorithm to count the amino acid level changes across the chromosome. And interestingly enough is to see that we can observe some off-target effect, because this is a parental strain, and you can see this is just the background, and there's no very obvious increase from some bias. But from the C to T, we can clearly see that there are more C to T off target, and the same here. But good thing is for this A to G base editor, the CRISPR A-best, there's no this kind of bias, and that means the A-best is more fidelity compared to C-best. And this is a deep analysis of this off-target, because we want to know if this off-target was happening inside of a gene, will cause some gene functions, or it just in some intergenetic area. That means maybe intergenetic area is not that important. And also we want to see that if this off target, like for example, this C to T inversion can cause some other stop codon introduction or sort of thing. But we can see from here, actually, the number is reasonable low compared to CRISPR-Cas9, because CRISPR-Cas9 will somehow cause genetic rearrangement, and that is like huge piece lost and sort of thing. So long story short is CRISPR best, at least based on the off-target evaluation, it is more fidelity than CRISPR-Cas9. Then the next step is that nowadays, if you want to do a metabolic engineering, during this one gene is normally not enough. So that means sometimes you need to simultaneously handling multiple genes. And for CRISPR system, there are many ways to do multiplexing. The most, let's say, straightforward way is to have multiple cassette of sgRNA. And multiple cassette, I mean, cassette means it has independent promoter, independent terminator. And just I mentioned before that for actinomyces, that will not a good choice, not be a good choice, because there are not many promoters and terminators for choosing. And if you keep using the same set of promoter and terminator, that will cause approximately instability, because if you use the same, exactly the same piece of fragment, it will somehow have some self-homologous recombination, so the proximity will be destroyed. And then we came up with a good idea, so we use this Cy4. So Cy4 is RNase, and it can specifically recognize this kind of RNA structure and have a cut. So that means if we just put them together, and in between we have this specific RNA sequence, so we can have sgRNA chain and just use one promoter to drive it. And after the transcript is there, so the Cy4, these RNAs can process the long RNA transcript to single piece of functional sgRNA. And then we also applied the Golden Gate assembly to do the assembly. So this is the 3-spacer 3 sgRNA case as a proof concept. So we target, one is the actinirodine, so the blue one and the red one, and then the other is this red one, and in between is the other famous compound CEA. So after that, we know that that the wild type, the parental strain, is blue, right? Because it has blue and red, and if blue is there, so red cannot be seen. But after this multiple targeting plasmid treat, we can see that there's no color at all. So it means the red is gone, blue is gone, and this one we didn't have color, but we need to use some other way to do the evaluation. For example, MS. But the take-home message here is the 3 spacer idea, or the Sci4 sgRNA self-processing idea worked. It's very good. That means we can simultaneously targeting multiple genes to do many other useful applications of metabolic engineering or even synthetic biology. So besides that, we also established sgRNA designer. So it's a Web 2, and you can just upload your own genome, and then you can just based on your own genome to customize, design different target genes, different sgRNAs, and also it has full support now for CRISPR-based application. For example, you can click just show the CRISPR-based output, or just show only stop codons mutations, for example. The star means that from Q can be introduce a stop codon here. So it is very nice, and you're welcome to try this system. So now it goes to the second summary, that this is the classic CRISPR-Cas9, and then this is the overview of the toolkit I developed, and we have CRISPR-Cas9, CRISPR-I, and also the CRISPR-Best, the base editing system. So now the other comparison is that if everything is optimal, to inactivate a gene using CRISPR-Best takes only around 1.5 weeks. So the time has been even shorter. So this is the overview of the CRISPR toolkit for actinomyces that I developed. So it has many different plasmid inside, but every single plasmid is one plasmid system. So it's very convenient. So from Cas9, we have 3 different plasmids, 3 different systems. One is that we can just take advantage of the native non-homolog engineering. So this incomplete, this defective homolog engineering pathway to generate a random size deletion library, or we can use this Lipids to reconstitute the non-homolog engineering pathway to have sort of precise genome editing tools to introduce small indels. And then if we provide the homology recombination template, so then we can use the cut and paste to have in-frame deletion or even knock-in, so insertion for some other purpose. And then if we use the Cas9 nickase plus the amynase, so we can have CRISPR-Best, CRISPR-C-Best, and CRISPR-A-Best to do the A to G converting, or C to T conversion. And the last part is that from the nickase, we can completely inactivate the Cas9 to make a dead Cas9. I mean, there's no nuclease activity at all. And using that, we can use this D-Cas9 sgRNA complex as a roadblock to just stop the movement of RNA polymerase. That means can have the knockdown phenomena of a gene. And because of this plasmid is a temperature sensitive plasmid, so it is a reversible system. And then we have some publications and we have some patents, and this system has been widely distributed worldwide. But this is not an end. It's just a start, because you will never know that there will be some even better tools coming out. And for sure, I believe that there are. So then comes to why I use CRISPR-Best, the name. So the name is just, sort of for marketing. And best is just best for now. And later it will be even more good tools and even better, best too. And then we hope that using all those higher ladders, advanced technology, advanced genome editing tools, we can bring the curve back. So we can have another golden age of natural products-based drug discovery, and to find a more useful, effective drugs to address to the drug resistant problem that nowadays we are facing. The last slide is for some acknowledgement. First, of course, Novo Nordisk Foundation for providing money for all the research I conducted, and also the group member from this NBC new bioactive compound section at CFB, and especially our PI, Professor Sandhya and Professor Tina Weber, and other group members. And now I'm happy to take any questions if you have some.
Dr. Tong, thank you for your wonderful presentation. I hope the audience has understood how much the CRISPR is very important to explore the hidden treasures. That is nothing but the natural products we can be able to generate from the various bacterial sources. You have given a lot of good information, and then interesting your findings about how that natural products are generated by using the CRISPR technology in actinomycetes. Our best wishes that you will get more findings in your new micro by using your CRISPR tool. Okay. Just I want to ask some of the questions posted by our audience.
Yes.
Dr. Kavita Tirumurugan from VIT, she has asked how the CRISPR-Best is precious to target the specific target gene without affecting the rest of the sequence.
That's a good question. So I will just go back to, basically I can just show you here. So the targeting, actually, that is the same as CRISPR-Cas9. Like, it is the same for all kinds of CRISPR-related application. So how to target a gene, that is defined by a piece of small RNA. So nowadays we call it sgRNA, so single guard RNA, and in single guard RNA we have a spacer, so just I showed here. So it is a 20 nucleotide in length, and that is coming from the target gene. And this piece of RNA will have like exactly complementary sequence to the target DNA. And that means if you put this system inside of your organism, then it will find the opposite strand of your target DNA and make complementary. That is how the targeting happening. And after they have this complementary, and that will cause some conformational change of Cas9 protein, and sort of activate the system. So either have a double strand break or single strand break. And then in the meantime, for the CRISPR-Best, so because we have a deaminase, and this deaminase is fused to the Cas9, in this case, Cas9 protein, basically. And then it will be allocated in the same position. And so, and because one strand of the double strand break, the target, has been bound to RNA, and then the other strand will be free, and that the free strand will be accessible to the deaminase, and deaminase can do the job. For example, if accidentally here, for example, the inside of the editing window, if there is a T, and this inside if there's C, and this C will be converted to a T, C to T, and then in the meantime, it will give you a stop codon. So that's how it works.
Thank you, Dr. Tong. Another one question. Is this CRISPR-Best system specific for actinomycetes, or it can be used for other system?
It is also a good question, and I can tell you that. So it should be a universal system. The only thing you need to do if you are going to use it for another system, the other systems, other different bacteria, other different organisms, you just need to know that if the plasmid, the repair count, is functional in your organisms or not. And that's one thing. And the other thing is that because later on you need to have the protein, functional protein, which means that if the codon is not optimal for your system, then you probably need to do some codon optimization. For example, you need to just based on your organisms codon usage table to redesign the protein sequence, the DNA sequence, and that's it. And besides that, I will not see any problems of using it, because eventually it is a protein, it is RNA, and RNA, protein, and DNA, they are just, every organism is the same. As long as you have a functional protein inside of your organisms, it should work.
Thank you, Dr. Tong. Is this technology can be used for the detection of COVID-19?
Sorry again?
Is this technology, this CRISPR technology,
yeah,
okay, can be used for the detection of COVID-19?
For degradation.
For detection.
Detection. Yes,
Yeah.
yes. Good question. So that will be not the topic that I present, but I can give you some more information for that, especially for targeting. So for detecting, for detection. So nowadays, many people, many groups, researchers, they are using CRISPR, CPF1. So maybe other people knowing as CPF1, but nowadays it has a new name, Cas12a. Just I had it here. So that is very interesting system. So it has here. So this one. So CPF1. So it also can give you a double strand break, but after this protein has been activated, so it has other activity. For example, it can cut single strand DNA, and then you can link many things to your single strand DNA for detection, right? So and then it can be used for detection of many different other things. If you can link your target to a single piece of olig, and this is for one application, and then the other application is other Cas protein. So it's ODAs coded C2C2, and that is RNA guided RNA targeting system. That means you can use it to detect many other like RNA virus and many other things. So yes, it is also a very hot topic or hot area using CRISPR to do the detection, even for small molecules.
Okay, thank you,
Yes.
thank you, Tong. Dr. Jebez, the time is there. Still, a lot of questions are coming.
So maybe
otherwise,
we can
wind up the session.
So we can take one more question if you want, and
Okay.
questions can be posted directly to the professor, and we can get the reply from him and send it to the participants.
Okay,
Yes,
okay.
I'm happy to do that, and I'm also happy to
Okay,
take the last
Tong.
question.
Just we will wind up with another one question. Okay. How does CRISPR-Cas9 system in bacteria recognize bacteriophage or viral DNA as lethal target?
So if I understand correctly of the question is saying that why the
CRISPR.
so how this system was coming from the original bacterial immune system, you mean?
No. How does
Yeah.
CRISPR-Cas9 system in
Yes.
bacteria
Yeah.
recognize bacteriophage or viral DNA as lethal target?
That means, okay, so that's a little bit complicated. So first, if we want to use CRISPR-Cas9 system as a genome editing, then the thing is that you have to know that in your bacteria there's no native CRISPR-Cas9 system, right? And because otherwise it will cause some so-called self-targeting. So because you know that at the very beginning, CRISPR-Cas9 system or CRISPR system is used as defense system or immune system for bacteria itself to stop the invasion of some alien DNA, some virus, right? And then so there's a very complicated mechanism for some bacteria to capture some virus or alien DNA fragment to integrate it into its own genome as like immune system as like spacer. And then but the bacteria itself will not trigger the expression of this spacer only if the second time the same piece of alien DNA or the same virus is going to infect the bacteria. So there will be some triggers to let the bacteria to respond to activate its expression of this part of the spacer, and then this spacer will form sgRNA, and then the sgRNA will form the complex with Cas9, and this Cas9 will go to attack the invader. So that's the immune system. And then later on is that people thought that, okay, if we can just stop the self-targeting, then we probably just use some other, like to use this system to other organisms without self-targeting, and we can provide some other repair mechanisms to repair the double-strand break. Even if like some bacteria, it can use as self-targeting, but in the meantime, you can just provide some repair template, for example, homologous recombination template, so it still can be used as a genome editing. So if that is the question you want to, maybe later if you don't like, you want more, then I can privately give you more answers for that.
Oh, thank you, Tong. Thank you for your excellent presentation. So that we hope that the audience got a lot of understanding about the CRISPR tool for generating various compounds from microbial source. Okay.
Thank you,
Uh,
and the pleasure is mine.
And then those questions which has not been answered, the audience, you can mail to Dr. Tong for getting the explanation. Thank you for your participation. We hope
Thank
that you got an excellent session. Thank you.
Thank you.
Thank you.
Thank you very much, sir, Rasul sir, for your wonderful sharing. So, now we are moving into the next session. We are delayed by 10 minutes actually. So we'll go ahead with the oral presentation. So the first presentation would be by Dr. Shashank Swain. So if you are ready, you
Yes,
can
sir.
go ahead with sharing your slides so that the oral presentation can start. Thank you, Rasul sir, once more. And thank you, Yajun Tong as well. And yeah, Dr. You can share your screen.
Sir, I'm unable to share because I'm talking in mobile, so I'm unable. Kindly share this slide from your side, and it is fine.
No, probably I don't have the slide. Dr. Selvarajan might be having 1 minute. Dr. Ramanathan sir, are you able to hear me?
Yes, Jabas, thank you. Please
Yes.
one more evaluator, Doctor.
Yes, so the other evaluator is Dr. Selvaraj.
Selvaraj. Okay. Whether he's on the session?
1 minute, sir. Dr. Shashank and
Yes.
evaluator, just if you can wait for 2 minutes, we are trying to put the other evaluator in. Just give me 2 minutes.
Okay, sir.
Okay.
Yes, the other evaluator is also there. Dr. Selvarajan, if you have the PPT, you can share, or else Dr. Shashank can carry on with the presentation, and the PPT could be shared to the evaluators later on, because we cannot delay any further.
Yeah, I don't have the PPT, sir.
Yes, Dr. Shashank, you can just start your presentation.
Okay, sir. Without slides, sir?
If you have the slides, you just go ahead with the slides. They'll understand with the concept, and once you send the slide, they'll start evaluating with the
But this
slides
is
as well.
the badge. It's unable to. Yeah, I'm not unable to. I'm unable to share.
It's okay.
Sir,
You
so,
can just
okay.
talk. You can just talk.
Okay, sir. Okay. Respected sir and my jury members and my dear colleagues, and I am Shashank Sekar from ICMI Regional Medical Research Center, Bhubaneswar, Odisha. I'm going to talk about synthesis of dapsone phytochemical hybrids against dapsone resistance Mycobacterium leprae, followed by a cost-effective computer-aided drug discovery approach. Yeah, nowadays leprosy is a chronic infectious disease caused by the acid-fast bacilli Mycobacterium leprae. And Mycobacterium leprae continue as the causative in public health problem worldwide, with the increasing rate of resistance of primary drug dapsone and rifampicin. And in last 10 years, record shows that in my slide, unable to visualize this slide or share this slide, but last 10 years, there is a highest number of Mycobacterium leprae cases were recorded from India, and now this rise of 1.2% per year is recorded. Because and other thing is Mycobacterium leprae is a chronic and in long-term morbidity in comparison to mortality rates. But in this case, in particularly in leprosy cases, there is a long-term morbidity and disability. This is the another national issue in nowadays, because there is in 2007 there is 3,400 cases for disability from recorded leprosy cases, but now in 2015 and 16 there is 5,857 cases, now 37% to 63% increase. If there is only drug available by that is MDT, multi-drug therapy is available for Mycobacterium leprae. But if this drug is really working, but how it is, the number of cases is increasing gradually in per day, in year by year. So there is some lacuna, because there is no drug available last 40 years for Mycobacterium leprae, and only MDT is used as a combination, different combination dose. So after that, you think about that, why this is dapsone. In MDT there is two or three drugs: rifampicin, dapsone, and clofazimine or ofloxacin. But dapsone is the primary drug used in the MDT. So dapsone is a sulfonamide group of class of drugs, antibacterial drug used for Mycobacterium leprae since 1937. So after mechanism of action, it is a competitor inhibitor of para-aminobenzic PABA, means this is bacterial enzyme, and another is sulfonamide, means dapsone. This is a competitor inhibitor of the target, that is DHPS, because dapsone target to DHPS. DHPS is a nodal enzyme in bacterial folic acid biosynthesis. So it is a competitive inhibitor of PABA. So after there is so molecular mechanism or molecular reports on the dapsone mechanism, and here it is clearly evident that in 53 position and 55 position there is a 309 and proline. Yes.
1 minute, your PPT is being displayed.
Okay, sir.
So you can have a look and you can tell the slide, and then you can start the presentation from the place where you have stopped.
Okay, sir.
Thank you.
Okay. And this is the mechanism, and this is mainly 53 position 309 and 55 position proline. There is a mutation. If there is mutation in 309 to alanine, isolation, and valine, and 55 position, it is the main cause of dapsone resistance. So this is from 2009 to 2015 molecular mechanism of action of dapsone and resistance pattern of the dapsone, and this is confirmed that 53 position and 55 position mutation is the major cause of adaptation resistance. Next slide, please. And this is the objective. Next slide. In objective, that is after that you define you are analyze that if you design your adaptation derivative, you can overcome this mutation, because everything nowadays it is modified after some days, because in bacterial cases that the nalidex-2 fluoroquinolone ciprofloxacin, that is the example of this how it is generated by year wise to overcome this antibacterial resistance. So this is my example. It is also example. This is a phytochemical conjugation, because all phytochemicals are some activity are recorded having some antibacterial, antioxidant, anti-cancer activity. But till now, there is no such type of phytochemical drug available in market in comparison to isolation, because there is so many phytochemicals are isolated. But till now, there is not utilization. This is a new concept. You hybridize a obsolete drug, means dapsone, and is a phytochemicals to develop a new hybrid drug against Mycobacterium leprae. And after that, you design a 5 to 7 5 suitable conjugates with dapsone with phytochemicals, then standardize. Because if you design a chemical, you synthesize, there is so many expensive required time and experiment. So either you used a computer-aided drug design, that is another plus point of this work, because you filtrate from the some parameter using some drugability parameters to select the which one is the best out of 5 designed hybrid drugs. Then you design after that you select a single compound to carry out an in vivo study. Then you can synthesize, then characterize the FTIR, UV-LCMS, then evaluate the anti-leprosy activity of the synthesized conjugate with the WHO guideline 0.0% of mg per kg. Because there is no another method, in vitro method is available for Mycobacterium leprae and activity testing. So only in vivo mice footpad propagation method is the only gold standard method till available in our clinical study. So investigate after that, investigate a host toxicity profile of active most conjugate using human lymphocyte isolator from umbilical cord plots. And basically this the mice footpad propagation method is carried out at Skiflin Institute of Leprosy Center, Tamil Nadu, Kadigari. Next slide, sir. And next slide, this is metal methods and reduce the cost during lead selection, reduce time during lead selection. Sir, next slide, please. This is method, because nowadays this how to reduce the time and how to reduce the cost in drug discovery. You can design, this is the conjugation. 4A, this is dapsone and hydroxychloroquine. 4B, dapsone with eugenol. 4C, dapsone salicylic acid. Dapsone with thymol. Dapsone with vanillin. And this is the schematic diagram of synthesized approach. This is the azo dye, because there is dapsone and another is in both sides of dapsone conjugated by through a linker, that is azo dye synthesized method of in medicinal chemistry. Then next slide, sir. Next slide, after design, there is some parameters, means computational analysis, that's prediction of activity spectra of substance, because DPC 1, 2, 3, 4, 5, and dapsone. There is a standard dapsone with design 5 conjugates, that is in this mycobacterium activity, anti-leprosy activity, anti-tuberculosis activity, toxicity class. This is a computational prediction and shows that DPC4, this is dapsone with phytochemical thymol. It is some more potent than others. This is green color. This is first, this one 0.745 and anti-leprosy also 0.74, means that this is the most active in comparison to other four high design conjugates. And this another is docking study, that is you can, because in previously you can already describe that this is due to mutation in this 53 position and 55 position. So you design these targets and mutate in 53 position and 55 position, then dock This is molecular docking approach. You dock each phytoconjugates in place of dapsone, and in means 6 dapsone including 5 conjugates. You dock each target protein in both wild type and mutant. And here also clear that this conjugate 4 also shows more potent than others. And here dapsone is minus 4.910. 910, but the dapsone 4 is minus 11. That is more comparison and more stability or more activity than dapsone after conjugation. And this is the next slide, sir. And this is the molecular docking study, how the interaction, how the conjugated drug interacted to wild type and mutant in 53 position and mutant in 55 position. And after that, because in computational is not good, it is a platform to reduce the time and cost, but it is not validated the original work. Or you cannot suggest that this is the main drug you can use. So
Dr. Shashank, you have only 2 minutes left.
Okay, this is another molecular dynamic study to stability. It is good or not. It is stabilized in this protein surface or not. This is the another aspect of the computational drug discovery. And next slide, sir. And this is the synthesis, because after computational study you can conclude that the dapsone 4 means conjugate 4 is the most potent compound. So you can synthesize the FTR. This is the FTR spectra, and this is the LCMS study, and this is the spectra of protein NMR, then surface visualization, the same test. Then next slide, sir. This after synthesize, you just carry out the DPC WHO guidelines of the mice footpad method in Karigiri. And so you can here it is this result of this dapsone and dapsone 4 conjugate 4, that is the dapsone, because really dapsone is used for the against a patient or give to a patient for 8 to 10 months, because our conjugated dapsone reduce significantly the growth rate of the bacilli. So it is clear that the after conjugation with the dapsone, it is reactivated, and it is active against Mycobacterium leprae, means dapsone resistance strain of Mycobacterium leprae. Next slide, sir. And this is the toxicity, because you are working in a medical environment. You can after a baby bath, after a bath of a baby, so you collect the cord blood, umbilical cord blood. You can isolate the lymphocytes, and it is another to use of the waste materials in medical waste material to a method to screening the active the toxicity story, that the lymphocyte treated with dapsone dapsone thymol conjugate and dapsone. There is in dapsone there is means it is not toxic. Generally, it is may be consumable after some validation, and this is the toxicity. Next slide, sir. And this is a summary of and future perspective. That is, phytochemical conjugation is a novel approach for possible reuse of clinical inactive antibacterials through suitable phytochemicals. Using advanced tools of bioinformatics and medicinal chemistry guided to synthesis most active lead chemicals, as well reduce the time and resources in current antibacterial drug development. Obtain computational ledger along with in vivo anti-leprosy activity and in vitro host toxicity outcomes prove that DPC for means dapsone and thymol conjugate could be used against dapsone-resistant Mycobacterium leprae and MDT in place of dapsone. Overall, phytochemical conjugation expanded to be an ideal approach for development of low toxic antibacterial hybrid chemical for control of MDR bacteria, as well as used reported phytochemical in mainstream medicine. The chemical conjugation could be employed for modification of other obsolete drugs in phytochemicals using tools of medicinal chemistry against any disease. And this is the outcome. It is in one publication in the cellular biochemistry and in recently in scientific reports of the study. And next slide, sir. And this is the acknowledgement. And so I acknowledge to my guide, my PhD guide, Professor Rabindranath Padhi, and Professor P. Sudhir Kumar Mayer, in specialist in medicinal chemistry. And special thanks to this Skipton Institute of Health Research and Leprosy Center, Tamil Nadu, India, for Dr. Sandeep Chaitanya, the research professor, and Professor Evangen from a director in this institute. And here, sophisticated analytical, that is CSI CSR Central Drug Research Institute, Lucknow, and Director Institute of Physics, Government of India, Bhubaneswar, and special thank to Department of Health Research, DHR, ICMR, Government of India for funding. Thank you, sir. Thank you. Sir, any question then again? Okay.
Yes, evaluators, it is open for questions.
Yeah, very good presentation, Shashank. Yeah. So can you just tell me what is the novelty of this work in just 2 sentences?
Sir, yes, sir. The first one is reuse of obsolete drug against this bacteria or the resistance bacteria, and second one is utilization of phytochemical in mainstream medicine. There is two novelty in this, and using in advanced computational bioinformatics or through including medicinal chemistry, reduce the time and cost in during selection or lead compound for synthesis.
Okay.
Shashank.
Yeah.
Yeah, Shashank, what is the docking and dynamics algorithm you used for your analysis?
Sir.
What is the molecular docking algorithm you have employed in your analysis?
Yes, sir. For molecular docking, that is Autodock software is used for the molecular dockings, and simulation that is means GLOMAX. Both are in free and both are used in Linux environment.
Okay. So first of all, you have published the data, so there is no question of like how you have finalized the compound, but but still I have a question like. You are saying from the docking score, some compound for DAPSON 4 is better than your DAPSON, something like that you are saying.
Oh, wait, sir.
Only with the binding energy, only with the docking score, you cannot say that it is.
Yes, yes. Because there another use, there is past prediction. That is how it is biologically active. This molecule after design, how it is biologically active. That's another tools use past prediction. And molecular docking, that is another, there is not, because due to short in 10 to 12 slide, you can not visualize all the parameters like Lipinski rule of 5, RO5, that is past prediction, predicted LD50 values, and there is so many parameters used according to drug discovery parameters.
My dear, before I ask the question, you are simply giving all the methodology again. My question is, binding energy is not only the criteria to different codes to go for the calculation of free energy. Did you calculate the free energy? Docking score is just a criteria to differentiate binders and the non-binders. But what about the free energy? That is very, very important too.
Yes, sir. Using MMPBS.
Yes, that is the validation for docking. Simply by docking score, you cannot claim that your compound is binding well, first of all.
Yes.
Okay.
Yes, sir.
But fortunately or unfortunately, your paper is published, so there is no question at all. But if it is not published, I will tell that this compound is not best. Okay. The reason is you are saying only docking score. Docking score cannot be acceptable.
No, sir. MMPBS is used for the individual.
But you didn't show MMPBS.
Yes, because. Okay, I'm not here because in this paper also described the MMPBS, but due to this minimize because there is the criteria for 10 to 12 flights, so unable to paste this.
But the second point is again, your data is too high for oral presentation, and it's very hectic to remember all the data. Given so much of data.
Yes, sir.
Maybe a small portion you can take it out and you can give it for presentation. Not like you start. I think you have given your complete PhD work. Is it?
Yes, so? Yes, sir. Yes, sir.
Complete PhD work. If you summarize and if you put all the figures in different order, you not even explained what parameter you analyzed by means of molecular dynamics. Tell me.
Molecular dynamics.
What parameters? What are all the parameters you have analyzed from molecular dynamics?
Umber tools use this molecular dynamics.
No. What are all the parameters you have analyzed by molecular dynamics?
Parameters means, sir?
Parameters means free energy, or
Van der, free energy score, free energy, then Van der score, then total means I think I'll show you this paper.
Where is the Van der Waals score? You didn't show any Van der Waals score and all.
Yeah, yes. This.
Do you show the Van der Waals score during the dynamics calculation?
Okay, I have this score, all score, because in this scientific paper,
you just tell me whatever you showed. Okay, what are all the parameters you are reported in the presentation slide? Can I know?
No, because all atom molecular dynamics simulation study, then trajectory analysis, MAPBS binding free energy. Then after that,
once again you are going to some different places. I am asking only in particular to molecular dynamics. What are all the parameters you have analyzed? Tell me. The slide is that. What are all the parameters you have analyzed? Tell me.
Okay, sir. Just a minute, because I'll show you this paper.
Yeah, the slide. No. I don't want paper, man. I want just a few information. Like the slide is there. 7th slide. It's perfect. Molecular dynamics graph. Everything looks good. There is no question at all. But what are the parameters you have used to conclude your compound is good? May you know?
The parameters in the Van der Waals energy.
Where is the Van der Waals? Where is the Van der Waals term here? See, that is why my suggestion. There is no Van der Waals graph at all here. Where is the Van der Waals graph? Okay, so that is why my suggestion.
Clearly.
There is no Van der Waals graph here in the screen. You have used different parameters, which is of not familiar to you, I guess. So my suggestion is, you can, during the presentation, restrict your slide to minimal number of content and give maximum insights within the data. Okay, that is my suggestion to you. Okay, but overall your presentation is very good. Okay, your data is very good. Thank you.
Thank you very much, Ramanathan sir and Dr. Selvarajan. So we'll go ahead with the next presenter. So it is Dr. S. Tyagarajan. So Dr. Tyagarajan, if you have your slide, probably you can share, or if you, oh yes. 10 minutes time, just stick on to the timing as we are going a little late.
Okay. Can you audible? Can you hear my voice?
Yes.
Yeah, thank you.
Be a little louder. Keep the mic a little closer.
Okay. Can you start?
Yes, please.
Thank you. Respected chair of the session and respected professors, teachers, faculty, scientists, and research scholar, and my dear students, very good evening to one and all present here. I'm happy to present my research work at this conference on natural product and synthetic biology. Myself, Tyagarajan. I did my work on the Department of Molecular Biology, School of Biological Sciences, Madurai, University, Tamil Nadu. Now I'm doing a postdoc research fellow in Department of Environmental Engineering, Harvard Institute of Technology, Vincent Quantum. So my research topic is isolation, identification, and functional characterization of delta-5 desaturated G microalgae. So as so far, microalgae have very beneficial role for human welfare, especially so their importance in production of protein, pigments, and polysaccharide biofuel. And now we are focusing on lipids, and especially PUFAs. PUFAs nothing but polyunsaturated fatty acid. Is the fatty acid containing 18 or more carbon chain and presence of 2 or more double bonds in the structure. So they are called as PUFAs. So they are classified as omega-3 and omega-6 based upon the presence of double bonds in their structures. So the major PUFAs are docosahexaenic acid (DHA), eicosapentaenoic acid (EPA), arachidonic acid (ARE). So these three fatty acids have highly beneficial for human development, growth, as well as metabolism. Especially DHA, docosahexaenic, are the majorly found in the brain membrane and retina of the eye. As well as DHA are major function in the infant and cognitive development of the adult age. Eicosapentaenoic acid and arachidonic acids are the two major fatty acids which involved for the production of eicosanoids and prostaglandin. These two molecules are involved in cell signaling and inflammation. Of course, these are the three major fatty acids which abundantly present in our immune system, regulatory system, reproductive system. So if the intake of these fatty acids are adequate level, we can regulate our cardiovascular system as well as we can protect our neurodegenerative disorders. Whether human can synthesis these two fast? Of course not, because human lacking delta 12 and delta 15 desaturated genes. So they must be obtained from the diet. So what are the major dietary sources where we can obtain these food fast for human purpose? It's plant oil and microalgae. Of course, we are taking a plant oil as a major source and fish as a secondary source, because all the vegetarian people can take a plant oil. What are the basic requirement of PUFA? So EPA, DHA are needed from the infant to the old adult people. So these are the different requirement of PUFA dosages provided by the World Health Organization, Food Agriculture Organization of USA, International Society for the Study of Fatty Acids and Lipids, European Food Safety Authority. For the adult people, it required at least 300 to 400 mg per gram in a day. But whether we are taking an adequate amount of these PUFAs in our daily basis, the question is no, because we are changing our food habitat. We are converting the modern diet to the Western diet. So our Western diet is drastically reducing the intake level of PUFA. If you are not taking these fatty acids adequately, we are facing a lot of diseases, especially neurodegenerative diseases like Alzheimer's, Parkinson's, and CVD, cardiovascular diseases, and aging, etc. What is the PUFA biosynthetic pathway? So whether plant can synthesize the essential fatty acids like EPA, DHA, and ARA, the question is no. The answer is no, because plants can restrict their PUFA biosynthesis until linolenic acid and alpha-linolenic acid. The rest of the metabolisms are absent in the plant system. So if you are taking the omega-3 and omega-6 fatty acids, we cannot get the essential PUFAs. But in case of microalgae, they are producing the PUFAs by de novo biosynthesis pathway. From the acetyl-CoA till the C22.6, the docosahexaenic acid produced by the microalgae by using series of desaturases and elangases genes. So my objective is, if microalgae can produce efficiently, but the outcome of microalgae for the commercial application is very limited. So then what are the possibilities to get the microalgae for the commercial outcome? So I have chosen a particular gene, delta 5 desaturase gene, to isolate from the microalgae and can incorporate in any of the heterologous system, can functionally produce these long chain fatty acids or not. Why I have chosen the gene for the heterologous system? Because currently the depletion of PUFA sources like fish and marine ecosystem are depleted. Continuous hatching of fish and pollution of marine ecosystem leads to the finding the alternative sources of PUFA. That's why we are choosing the microalgae as a potential sources for the isolating of the PUFA biosynthetic genes. The microalgae are the primary producers in the aquatic systems. Of course, they are having a several desaturases and elangases gene to produce the essential polyunsaturated fatty acids. So the delta-5 desaturase is the key enzyme to produce the C20 long chain fatty acids like eicosapentaenoic acid and arachidonic acid. So that's why we have used a genetic engineering approach to incorporate the delta-5 desaturase gene to the heterologous system to produce high-value lipids for the pharmaceutical and nutraceutical application. So for my studies, I have chosen isocasis species. Isocasis species is very well known and enriched with high amount of DPA and DPA and DHA. So we have chosen isocasis for my study, and we have cultivated in our laboratory in Valley medium. After 7 days, we have isolated the RNA using Pragasam method. Then we have synthesized the cDNA. From the cDNA, we isolated the delta-5 desaturase gene using gene-specific amplification. And finally, we found nearly 1.1 kb of delta-5 desaturase gene from microalgae isocasis. Further, we want to confirm the isolated gene is belong to delta-5 desaturase or not. So the gene was further cloned into pGMT by precision digestion, which showed the release of 1.1 kb size. And this was further sequenced, subjected for the sequencing, and we found nearly 1,170 base pair of delta 5 desaturase gene. We found the nucleotide blast showed 96% similarity to the isocasis vacuna. So further, these sequences was subjected to the multiple sequence alignment to confirm whether the isolated gene is delta 5 desaturase or not. Of course, with the available other desaturase gene, with the available of our sequences, we identify the conserved histidine domain of delta 5 desaturase gene and the cytochrome B5, the electron donor domain, which was found in our sequences also.
Dr. Tyagarajan, another 2 minutes left.
Yes. So then further we find out the ORF region and we identify the 1145 region as a ORF region. I have taken this ORF region for further protein expression. The protein expression was done by PGX42 vector and the transformation was confirmed by restriction digestion and it was further expression with the PGX42 vector. By using 1 millimole of IPTG, we have found the overexpressed protein about 70 kilodalton. The GST has a 26 kilodalton size and our expected protein size is 54 kilodalton size. The fusion protein was found at the 70 kilodalton size. Actually, equally can restrict their fatty acid synthesis up to C18:10 fatty acid like C20 and C22. Our ultimate aim is equally want to produce C20 fatty acids like EP and DHA. So the respective desaturase gene substrate was additionally provided to the medium. So we have provided the substrate like DGLA, docosahexaenic acid, and ETR, eicosapentaenic acid, to the both wild and recombinant E. coli. And we have analyzed the gas GCMS, and we find the successful production of new novel fatty acids like EPA and ARA, the recombinant system, which does not found in the wild. And this was my summary of conclusion of this one. And we have isolated the novel gene, which never ever isolated from the isocasis species. And we are the first one, we isolated the isocasis species and we submitted in the NCB accession number. The isolated gene was confirmed by delta 5 desaturase using different bioinformatic analysis, as well as the desired protein expression was also found in the E. coli PGX42 vector. The functional characterization also successfully produce the eicosapentaenic acid and arachidonic acid in E. coli. And this is the preliminary studies, and we need to do a further experiment want to confirm whether the E. coli can produce the long chain fatty acids, which previously I have showed the different genes like delta desaturation elongases gene, why we can incorporate those genes in E. coli and produce E. coli as alternative organism. Actually, there are previous studies have already reported the synthesis of omega-3 and omega-6 fatty acids in plants as well as in yeast, but there is no study reported in E. coli. So this is the first study. We hope that we have successfully attempted and produced a long chain fatty acid. So I would like to thank my guide Dr.K.Sathiyanarayanan and for giving the wonderful opportunity to do my PhD and successfully completed. I thank DST sir for financial support. I thank DBT IPLS, NSCBS, GCCES, MIPS, EAGS for providing, and SBS MKU for providing their facilities. And this is the origin of evolution. So man were evaluated from the monkeys, but still we are changing our food diet. That's why we are looking for the pharmaceutical and nutraceutical application. So at least we should take short amounts of omega-3 fatty acid in our diet, especially 250 to 500 mg, especially for Indians. They should take in diet to avoid PUFA deficiency disease. Thank you. Thank you very much, sir.
Fantastic, Tyagarajan. The presentation is good. I think Selvam will ask questions.
Thank you, sir. A very good presentation, Dr. Tyagarajan. Yeah. So I have some few suggestions and some queries. So whether this one is this work was published already?
No, sir, not published. Not published.
Okay. Yeah, I have seen a similar work in applied biochemistry and biotechnology.
Yes,
so that is why I'm asking you whether you have published or not.
No, sir. That is my another work. I have only published the applied biochemistry and biotechnology.
Okay.
That I have taken from different algae and different genes, not this gene.
Okay, so you have done
in our laboratory, we are constructing the proof of biosynthetic pathway in E. coli. So, as I showed in my slides, there are 6 and 7 desaturated gene involved in proof of biosynthesis. So, this is one among the enzymes which now I presented.
Yeah. So some of your agarose gel and some other photographs you showed, there is no scale actually. And
So this one?
yeah, before
Actually,
that.
I'm just representing the size of the vector and the insert. I'm not showing the ladder marker.
Okay, so what is that sixth lane?
The 6th lane is a wild one.
Okay, so why that band is actually shear actually?
While isolating the plasmid, there is some mistake. That's why after it was a shearing because of RNA scale.
Okay, so what percentage gel used?
This is 1%.
1%. Okay, so can you go back? Can you go back? Little bit, yeah. Yeah. So here under like, what is this picture you have given? This one.
So the first one is isobasis. So this is the
Okay.
microalgae we are cultivating in our laboratory. The
So
microscopic here.
yeah. So
And the
microscopic.
second one. Okay.
Yeah. So microscopic image should have been some scale. So whether it
Sir,
is
actually,
taken in 48.
actually, the compound microscope available from our lab, there is no computerized microscope. So that's why there is no scale.
Okay. So this one, the second one.
Sir, second one. The second one is a PCR amplification of delta 5 desaturated gene from cDNA.
Yeah, but there is no legend, right? So lane 1, what is lane 2, what is lane 3? Whether you have duplicated this one or what is it?
So all are same. Sir.
Same. So okay.
I have taken the cDNA in the different vials. Then after the PCR, I just confirming whether I'm getting amplification or not.
Why you use this GST tag?
The GC tag, I'm using a PGX42 vector.
Okay, so whether you're getting the protein, whatever you are checking.
Yes, sir. Yes, sir. So this I have shown another protein expression vector. So the first lane is a wild vector. The lane 2, 3 is different transformation colonies. So among 2 from 3, 4, 5, 6, we got a overexpressed protein, almost at nearly 70 kilodalton size. So the 70 kilodalton is our expected size. So that's why we confirm that the protein is expressing in transformed E. coli, and further we move for the functional capacity.
Whether you check for the inclusion bodies?
No, sir, there is no inclusion bodies. Since if I have checked with the PET system, there is no expression in the PET. Maybe because of inclusion bodies, I couldn't get the expression in the PET system. So that's why I changed the expression system, like PGX42. So then we got with the GST.
Okay. Okay, thank you. Thank you.
Thank you.
Thank you very much, evaluators. The next presenter is Moni. We are running short of time, so don't, unless and until the methodology is novel, you do not have to explain the methodology in detail. So you can directly, if it is a novel protocol, you can explain the protocol. If not, directly go ahead, just tell the protocol and go ahead with the results. Okay, thank you.
Moni Philip, are you in the screen? Are you in the session?
Let me see here.
And why you are not starting your presentation?
She is
Any issue?
messaging.
Moni?
She has to check the microphone, I think.
Connected audio to phone.
Okay, just give me another minute.
Good afternoon, sir. Are you able to hear me?
Yeah. Please.
Okay. Good afternoon, all. My topic of presentation is comparative in silico investigation of phytocompounds from Nilavembu-Kudineer formulation. So basically, this idea for this study came up when I read a newspaper article when the Tamil Nadu government was supplying this particular Nilavembu-Kudineer to people in Chennai, especially when they were affected with this coronavirus. So we had an idea, what if we can try exploring the compounds that are present, and we just did a docking analysis to see if these compounds are actually having any interaction with the active site of the SARS coronavirus. And it is just a preliminary study. We are now performing the next phase, which is our molecular dynamics and all. So here, this presentation is just on the preliminary docking studies that we had performed. So first of all, what is this Nilavembu-Kudineer? So basically, this Nilavembu-Kudineer is a herbal formulation. So it is actually composed of 9 different drugs.
And that is fine. Moni,
yes,
Moni Philip.
Sir, yes, sir.
Moni Philip.
Yes, sir.
Yeah, this is fine. Nilavembu-Kudineer is fine. You just go to the next slide.
Okay, sir, fine. So we thought, okay, since it was used in previous viral diseases like chikungunya and dengue, we thought, what if it's used in SARS corona? So here we were targeting the specifically on the protease. So the role of protease in corona is basically it is going to cleave the the proteins that were translated from the genome RNA. So this forms a major it has a major application in which if this main protease is going to cleave the entire protein which is translated, unless and otherwise it is cleaving, the SARS coronavirus cannot make functional proteins. So if we target this particular main protease, there is a chance that this main protease could be inhibited in a way that the translated protein cannot be cleaved into functional proteins, so that the replication of the SARS coronavirus can be limited. For this, basically what we did was we purchased this Nilavembu-Kudineer formulation from a local Siddha shop, and we tried extracting the phytochemicals using ethanol. Then after the extraction of phytochemicals, we just did a GCMS analysis to find what are the different compounds that could be possibly present. So from that, we had a cutoff between 60 to 600 kilodalton. From that, what we found was we were able to find around 8 compounds. And what we thought was, let's do a simple investigation on how these compounds are acting on the active site of the coronavirus main protease. So what we did basically was we took the complete genome of SARS coronavirus from NCBI database. Then we did gene annotation, and then we blasted it. We used a P-blast. Basically, it is a protein blast. So after doing the blast, we were able to find certain similar sequences. And among the sequences, we were able to find one with 100% similarity. So that particular sequence was already submitted in PDB database, Protein Data Bank. So we took that particular sequence as a template to develop model. So what we did was basically from the annotated gene, whatever we had, we made a homology modeling using modeler. So after we did this homology modeling with modeler, we actually
Moni Philip.
Yes, sir.
If it is 100% similarity, then why you are going for modeling again?
No, sir, we just wanted to check what the model is.
What is this? Yeah, the genome sequence is 100% similar. That means that is a protein structure. Then why you are going for modeling?
No, sir, we just want to do a study. It was part of a student work, yes, sir. So then we did a structure validation. Then what we did before docking, we did a minimization of energy of both protein as well as ligands. Using proteins, we're using SPDB software, and ligands we energy minimize using Avogadro software. After which we did docking analysis using Autodoc Veena. So then this was our modeled protein, and it had an RMST of around 0.258 Armstrong. So after that, we took this protein, as I told, for docking analysis. So this is basically the Ramachandran plot.
You go, Moni Flip.
Yes,
again,
sir.
I'm sorry.
I,
You go taken 100% similar structure.
yes, sir.
And you go modeled.
Yes, sir.
But after modeling, you ends with the RMST variation of 0.2. This is absolutely wrong. See, RMSD will be higher only if you are not getting similar structure during the database search.
Okay.
If you are getting similar structure, how during the modeling, how the RMSD will vary? RMSD will go more than 0.0. It's supposed to be RMSD is supposed to be 0.0.
Yes, sir.
Because it is 100% similar structure, you know.
Yes, yes, sir.
How it is 0.258? Do you know 0.2 means it is there will be a great variation. That is a significant variation, my dear.
Oh. No, sir. But when we did, we got.
See, my question is 100% similar.
Yes,
is there any one residue is different? So
sir, but,
with the 100, tell me, go ahead.
No, sir. Continue.
With the 100% similar sequence, you are doing modeling study, and you are saying the modeled structure is having RMSD of 0.258. It is unbelievable. What do you know? What Swiss model will do?
Sir, we used modeler for.
Okay, what modeler do you know? What modeler will do? Homology modeling using modeler. What do you know? What modeler will do? It will replace the existing template amino acids by your sequence amino acids and give you as a output.
Okay, sir.
It will take the main chain, it will take the backbone and replace that amino acid by the amino acid which is there in your sequence.
Yes, sir.
Okay. So ultimately, during this replacement, there is no major change.
Okay, sir.
So it will give us the same structure. So now your work is you have to take that particular output structure and you have to superimpose with the existing structure. And if you superimpose, both structures are same. How the structures may be different?
But I don't know, sir. When we actually did the overlay, there was a slight difference.
Okay, go to the next slide.
So yes, sir. So basically, we did this docking analysis, sir. So for this, we took two standard drugs, antiviral drugs, favipiravir and nelfinavir. Then we also took a PDB structure also, just in case to compare our results. So
we used this nelfinavir as a reference.
Sir, nelfinavir was previously showed as a antiviral drug, which was targeting some protease. So we thought, okay, we just wanted to see.
Some protease, that is not your coronavirus protease.
No, not coronavirus protease. Just
then how you can take nelfinavir as a reference?
No, sir. We just search for antiviral proteins, antiviral drugs which can target protease. So nelfinavir is a protease inhibitor, basically for retroviral drugs. So.
Okay, just my suggestion. Is there any other you want to say? You go to the next slide.
So next slide is basically the docking. Then we did
next slide. Next slide.
ADME properties and toxicity analysis. That's all.
So yeah, just for your suggestion.
Yes,
don't show this homology modeling, sequence analysis, sequence comparison, and all the other stuff. Straight away, first of all, structure is available, so you can take that structure.
Okay, sir.
Okay. And you can take all the compound, whatever the compound is isolated from Nilavemba, all the compounds you can take, and you can straight away go for docking using blind docking mode.
Blind, okay.
You no need to take any reference molecule because there is no proven drug. So in that case, you no need to take any reference molecule.
Okay, sir.
Okay. Moreover, you should not dock only with the main protease, because there is no guarantee that your compound will attack only main protease for the management of coronavirus. It may attack any protein present in the coronavirus.
Yes, sir. We actually docked with different RNA-dependent RNA polymerase and so many different NSPs also.
There are some 6 targets or few targets are available.
Yes, sir.
RDRP, MPRO, the main protease, then 3C-like protease. There are so many targets that are available. So my suggestion is you identify the structure.
Okay, sir.
Those structure you dock with all the targets.
Okay, sir.
Don't compare it with reference.
Okay. Just do a blind
that
docking.
may have blind docking.
Okay, sir.
Among the compounds, if you say this compound score is better,
okay,
then it may have some meaning. Just an advice for you.
Yes,
okay,
yes,
don't
sure,
take it in negative sense. Are
no,
you
no,
assistant professor?
Sir. Yeah, now I'm assistant professor.
Okay. So you are an assistant professor, So you just tell them to your students.
Okay.
No need to compare it. This will be a blind docking. At least this data would be useful. Okay. Otherwise, this data is of no meaning. RMSD data is not useful. Modeling data is not useful. Okay. Afterwards, you can compare to nilfinavir, flavipravir. That is also not useful because that is not the drug of choice. Okay. Ultimately, you are targeting only MPRO. That is also not useful.
Okay.
Okay.
Yes, sir. Sure.
Keep it in mind. Thank you.
Okay, sir. Thank you for your suggestion.
Selvam, sir.
Yeah. No,
okay.
Sir.
Thank you very much, the evaluators. So now, actually, we have 2 more participants, 2 more presentations. They have not replied to the message. So Hashim A.
Well, that's fantastic.
1 minute, sir. So Hashim and Mrs. T. Sheila, both of them have not responded as of now. So once they respond, I'll revert back to you. Thank you very much for the evaluation. Thank you, Ramanathan, sir, and thank you, Dr.
Thank you so much.
Thank you very much.
Care.
Thank you, sir. So,
so
we are moving ahead with the next
much.
session. Thank you, sir. And we are moving ahead with the next session, for which yes. So I request Dr. P.K. Suresh. He's a professor working at VIT. So take over the next session as he is the chairperson for the next session. Over to you, Suresh, sir. Thank you very much.
Hello. Can you hear me?
Yes, sir. Yeah.
Yes, so we can
Hello,
hear you.
can you hear me? Hello, can you hear me?
Sir, your voice is echoing.
You have to unmute. You have to unmute me here. Please unmute here. Can you unmute?
1 minute, sir.
Jabez, can you unmute me?
Yes, sir. 1 minute.
Here, right here.
Yes, sir.
You have not. Hello, can you?
Yes.
Can you hear me?
Yes, we can hear you.
Yeah, Jabez, could you unmute the other one here, right? The other screen. Can you unmute this? Can you unmute?
1 minute, sir.
Okay, in that case, can you project the profile of Dr. Apu Ratnave?
Yes, sir.
Okay, please. First of all, I would like to convey our heartfelt thanks to Dr. Apu Ratnavelu for having kindly consented to deliver a talk today. I want to thank you on behalf of the organizers. Okay, Dr. Apu Ratnavelu is currently the Associate Dean, Institutional Planning and Faculty Development, and Executive Director, Ramwag Goodwin Institute for Cancer Research. Dr. Ratnavelu initiated his research efforts focusing on discovering new therapeutic targets and novel drugs for cancer treatment. He has published nearly 50 peer-reviewed research articles, co-authored a textbook, written 2 book chapters, and presented more than 75 scientific papers in national and international conferences. He also serves in the editorial board of several esteemed scientific journals and has conducted peer review of several scientific manuscripts for publications. Dr. Ratnavelu has received 4 patents and is currently planning to perform preclinical trials for his anti-angiogenic drugs with pharmaceutical companies. His primary research focuses on discovering novel methods for predicting treatment outcomes and developing drug combinations that will be useful for more potent and less toxic treatment of cancers. In this regard, he is involved in identifying gene expression profiles that predispose individuals to certain types of cancers. He has also developed novel methodologies for identifying kinases and phosphatases in biological samples. We are very glad and happy to have a scientist of eminence in our midst today to deliver a talk amidst the pandemic-related lockdown. His talk is entitled Use of Phycocyanin and Other Natural Products for Cancer Treatment. Dr. Apu Ratnavelu, you have 45 minutes from now to deliver your talk. Just 2 minutes before the end, I will remind you that you have 2 minutes left. After the talk is over, I will select a few questions from the chat box, the Q&A section, and will pose them to you. And the remaining questions will be mailed to you if we have to cut short the Q&A due to time reasons. Dr. Apu Ratnavelu.
Thank you, Dr. Suresh. Can you hear me well?
Yes, sir.
Thank you, sir. Thank you for a beautiful introduction. Also, I would like to thank Dr. Karthik Loganathan and other members of the organizing committee for inviting me to participate in this seminar symposium. It's a pleasure to be here. It's 7:15 a.m. here. I started joining this symposium around 6 o'clock, and I feel a little bit sleepy. So forgive me if I speak during the presentation. So I will try to share the screen and start with a brief introduction of our institution and then go from there. So let me share the screen. Are you able to see the slides? I hope you are. I'm not able to share the screen from here. So let me try again.
Sir, please open the PPT first, PowerPoint, and then share the screen, sir.
Yes, it's open. The PPT is open. So I'm trying to share through the Zoom.
Yes, the green share button. And then
Yes. It says one participant can share at a time. So I'm clicking on that, and then the screen is not coming up. I've done this many times, and unfortunately, I don't know what is happening now.
Just give me a minute, sir. I'll just check.
So while we are waiting for the screen to come up, maybe I can continue to talk. I am from Nova Southeastern University, which is one of the private institutions in Florida, one of the largest private institutions. We have a total of 18 colleges, and within the health professions division, we have 7 colleges. Probably we are the only second university in the country, in the US, with 2 medical schools offering MD and DO programs. So we graduate nearly 3,000 doctors every year. I'm giving you these details to tell you, even though we are small, we are growing. The Ramba Goodwin Institute for Cancer Research is one of the research units of Nova Southeastern University. We are engaged in cancer research. My research started, I would say, in '93, cancer research with NSU. And as Dr. Suresh mentioned, we have obtained not just 4 patents in the last 3 years, we have added 3 more patents. So we have 7 patents for small molecules, which are targeting angiogenesis. These are anti-angiogenic compounds. We are waiting for partners to collaborate with us so that we can do the clinical trials. So this is to show, let me try to share the screen again. It says one participant share the screen, and I'm unable to.
So you must be able to share the screen now.
Now? Okay.
Please
try again. Okay.
Yes.
So share the screen. Okay, it's not giving me the option to select the screen, unfortunately.
So click on the center of that share screen.
Share screen, yes, I'm clicking on that.
Click on the center of the share screen.
Okay, got it. Thank you for your, okay, now I got it. So I talked about the university. So can you see my slides now?
Yes, sir, we are able to see it.
All right, thank you so much. So this is one of the units, one of the centers of the Nova Southeastern University. And through innovative research, we want to develop new drugs and cure cancer. That's one of our goals. Recently I was looking at the number of COVID patients around the world. It's about 11 million. And if you look at the number of cancer cases around the world, it's 18.1 million. It grows, even though a couple of years ago, there was big news about decline in cancer incidence in the US. Hopefully we can see that in the rest of the world. When you go to MD Anderson Cancer Center, in most of the buildings, they have stricken out the name cancer, saying that we have conquered cancer. Now, in my opinion, we're not there yet. We will be there maybe in the next 10, 15 years, but conquering cancer is coming close to us, but we have to do our job to reach that goal. So as part of that effort, our institute is focusing on drug discovery, translational research, genomics, pathway analysis research, and we are also developing recombinant proteins that can target various cancers. And today's talk is going to be focusing on one of the natural products that we have tested in our research labs. We have tested the effects of resveratrol, genistein, curcumin, Tulsi extracts, and also phycocyanin. Today's talk, I'm going to focus on phycocyanins. And as Dr. Suresh mentioned, we have several patents. Many of them are international patents, and we are in the process of now, starting the clinical trials in collaboration with the local companies. And if anybody from India wants to participate, we can really work together. Glioblastoma is one of our focuses because, this famous senator died because of glioblastoma, which created a lot of awareness. Since our compounds are targeting angiogenesis process, we are trying to use our compounds similar to Avastin for treating glioblastoma. It shows good brain delivery, and also Uma, who is participating in this seminar today, and also Dr. Tyagarajan, they are doing some of the analysis to confirm the effects of our compounds, F16 and JFD, for treating glioblastoma. So, and in addition to that, we are also looking at some of the side effects caused by chemotherapeutic agents. And that's one of the reasons why we are testing natural products. Basically, by using smart combinations, many of the drugs have come from plants and other living organisms, such as Taxol, topotecan, vincristine, vinblastine. But these are therapeutic compounds with a lot of pharmacological benefits. But our goal is to take the phytochemicals with nutritional value, antioxidant properties, and see whether we can really supplement the therapeutic effects of the chemotherapeutic agents. A lot of patients take supplements, dietary supplements, once the cancer starts. But our goal is to know the risks for developing cancer and start with these phytochemicals before the cancer starts and put the cancer on hold. And if the cancer starts, now use these phytochemicals in combination with the drugs such as topotecan, Taxol, and minimize the side effects. As you can see, peripheral neuropathy is one of the side effects that comes with chemotherapy. And our goal is to use combinations with the phytochemicals and minimize the side effects. This way, you are not only extending the therapeutic benefits and also minimizing any complications that may be coming along with the chemotherapy-induced neuropathy type of side effects. So, we are also doing drug discovery on the fast lane. Basically, as Dr. Ramanathan and other presenters were showing, they are doing a lot of docking studies, not myself, my collaborators. They are doing a lot of docking studies of the FDA approved, those drugs which are in clinical use, and see whether we can use them for treating cancers. By doing this, we are not only saving time, we are going to be saving a lot of money. For example, if it is taking 6 years, $300 million to bring a drug to clinical trials, by doing this repurposing, it is called repurposing, we can shorten the time and also save a lot of money in bringing an existing drug to patient's bedside. So this is one of our focuses. So some of the examples, for example, the arthritis drug, leflunomide, is used for treating prostate cancer. Dapsone can be used for treating Kaposi sarcoma. So these are some of the approaches we are utilizing at this point of time. Antioxidants are used not only by healthy individuals, and it is used by a lot of cancer patients because it can cause free radicals, can cause base modifications, and these base modifications can create effects similar to gene mutations and leading to cancer development. And our goal was to not only stop the production of the antioxidants by blocking the effect of the carcinogenic chemicals with the target tissues, and also minimize the process of induction of damages. As part of that effort, we are looking for phytochemicals from natural sources, antioxidants we are looking for from the natural sources, and we are looking for complex carbohydrates from natural sources. As one of the presenters, Dr. Tyagarajan, mentioned, we are looking for omega-3 fatty acids from natural sources. All these are to maintain healthy life and also minimize the risk. Omega-3 fatty acids, I'm going to skip these slides for the benefit of time, are good in terms of providing support for maintaining healthy body. So one of our earliest studies were done with Tulsi Extracts, and this should pave the foundation for the Phycocyanin study I'm going to be presenting. Since a lot of people are coming into the waiting room, the screen is kind of coming up in the middle of the presentation, and I hope it won't be interfering in others' view. So when we looked at the effect of the Tulsi extract, our goal was to see whether Tulsi extract can systematically induce programmed cell death in cancer cells. And okay, hold on. Okay, so we selected LINCAP prostate cancer cells, and we did a dose response study. As you can see, the cells, when they were treated for 48 hours, we were able to see cell death with 100 microgram per ml of the ethanolic extract. 100 milligrams per 100 micrograms per ml is a kind of a heavy dose, but that was really for looking at the individual effect of the extract coming from the Tulsi. And we were able to establish the dose response curve for 24 to 48 hours. And we were able to see good effect in 48 hours. Of course, in 24 hours, there were some cell death, not to the significant level. And flow cytometry analysis also showed that this 100 microgram per ml of ethanolic extract can induce cell death. But that was not our original goal. Our original goal was to compare and also combine with drugs such as topotecan. For example, the level of cell death that was seen with the 5 micromolar, as you can see, is very high compared to 100 microgram per ml of the ethanolic extract. But we were also able to do the fluorescence analysis. The propidium iodide binding study showed that both ethanolic extract and topotecan were able to induce cell death in these prostate cancer cell lines. So, and in addition to that, when we measured the caspase activation, both caspase 3 and caspase 9 activities were elevated with these treatments, and particularly with the ethanolic extract treatment. And that showed that the intrinsic pathway is activated. When you look at the apoptotic mechanisms, there are 2 pathways that are involved. One is the intrinsic pathway, the second one is the extrinsic pathway. This ethanolic extract coming from Tulsi seemed to be inducing the intrinsic pathway and leading to PARP cleavage, and that is what you are seeing here in this slide. In addition to cleaving the PARP, which is the DNA repair enzyme, in addition to cleaving PARP, the BCL2 level was also decreased by the ethanolic extract, which confirmed that both by decreasing the levels of BCL2 and also by activating the intrinsic pathway through elevation of caspase 3 and 9, caspase 9 and 3, Tulsi extract could induce cell death. So this kind of established our system for doing analysis with other compounds. This is when my collaborator, Dr. Miro Gantar, from one of the neighboring universities, came to me for collaboration. He was focusing on extracting phycocyanin from the blue-green algae that was growing in the Everglades. And I had a picture of the Everglades, but it looks like it is not appearing here. And if it appears, then I will talk about it for a few seconds. So this particular, let me close my screen 1 second. Okay, all right. So hopefully this, so this extract, the phycocyanin extract was, tested by Miro for other purposes, but he came to me for collaboration. And he wanted to, this is the Everglades. So the Everglades is very close to where we are in Florida, South Florida. And basically, I would say within a few minutes from where I live. And it has a lot of living, it's a natural habitat for a lot of living organisms. And, it has marshlands. And this is where the blue-green algae grows. And Miro was able to extract 2 strains of the blue-green algae, Limnothrix and spirulina, and he was able to extract phycocyanin. He wanted to see, even though there were many reports related to the effects of phycocyanin in stopping cell cycle progression and inducing cell death, he wanted to do systematic studies with our collaboration to determine whether phycocyanin can induce cell death in cancer cells. So as you can see here, phycocyanin, we prepared 1 micromolar solution, 10 micromolar solution. As expected, 10 micromolar was giving more than 60% cell death. I'm sorry, this is a topotecan. This one I was pointing at topotecan. So 10 micromolar topotecan was giving more than 60% cell death, but phycocyanin was not able to really show that much of cell death, which was not bad. But when we combined phycocyanin, the screen, let me minimize the screen. Okay, when we combined 500 microgram of phycocyanin with 1 micromolar topotecan, we were able to potentiate the effect of the phycocyanin. I'm sorry, the topotecan. So if you see the topotecan 1 micromolar, it was giving about 75% cell death. by combining with phycocyanin, we were able to really enhance the therapeutic benefit coming from topotecan. So the idea is by using combinations like this, we should be able to reduce the dose because higher dose produce side effects such as peripheral neuropathy. And by reducing the side effect, maintaining the therapeutic benefit, we would be able to treat the patient more successfully and that they can continue with the treatment without going through the drug holidays. So this was the idea. And we were able to also look at the caspase activations, the combination, which is shown in the kind of the light purple bar, was able to show higher activities of the caspases. In addition to showing let me move the screen because this pop-up screen is blocking the blots. So as you can see that the BAX levels were elevated in combination treatments and BCL2 was lowered. So, and cleaved caspase 3 activity or level was very much higher in the combination treatment. So these are individual treatments. As you can see, 500 microgram of phycocyanin did elevate the activities of the caspase 3, but the combination was working much better. This is with 1 micromolar topotecan, and of course with phycocyanin. So, this is a perfect example of something that can be taken as a dietary supplement if it is combined with right therapeutic agents such as topotecan, which is known to induce programmed cell death. It can, actually augment the effects of the topotecan by decreasing the levels of the BCL2 and elevating the levels of BAX and activating the intrinsic pathway. So let me move on to the next slide. So I'm trying to move on to the next slide, and it's kind of frozen. So let me see. Okay, all right. So PARP cleavage was also seen in these treatments, which confirmed that this combination can work in the prostate cancer cell line. And subsequently, we were testing, looking at the DNA fragmentation. This is one of the hallmarks of the apoptosis induction, where the DNA is fragmented, leading to completion of the cell death. So, at that point of time, Florida Department of Health released an announcement saying that, if we can use any of the natural products for treating lung cancers, we would be able to fund. So we started with the testing use the lung cancer cell line A55459 cell lines, and we were able to get some funding. When we looked at the gene expression profile by using the RT-PCR array, we were able to see elevation of the BAX in the array analysis also. In addition to that, we were able to see elevation of GAD45 and a few other genes, which confirmed that phycocyanin by itself can induce the expression of certain genes. And this was done with the lung cancer cell line, and the previous data I showed was for prostate cancer cell line. But the data you are seeing here is for the lung cancer cell line, which looked very similar to what we saw with the prostate cancer cell line. The combination of phycocyanin and topotecan 1 micromolar did the same thing. It was able to decrease the levels of the BCL2, elevated the levels of BAX, and that led to the cleavage of the PARP and completion of the cell death. So now the question was, this is a big molecule. Phycocyanin is a large molecule. I don't remember the molecular weight. Maybe Mira would be able to tell me the molecular weight immediately. It's a huge molecule. So we were wondering whether this molecule is able to really penetrate the cells. So this was a big question. And because this question was asked by many other reviewers, because huge molecules getting into the cells is not that easy unless you make pores by using detergents or some other alcoholic treatment. So when we did the binding study, fluorescence binding study, phycocyanin by nature, it can fluoresce. And when we were able to do the binding studies initially, after 1 wash, it was showing binding to the plasma membrane. As we were increasing the number of washes, after 2 washes, as you can see, the binding was decreasing, and after 3 washes, completely washed out. So that showed that the molecule is not entering, if the molecule is entering into the cells, it should be able to really stay inside of the cytoplasm. But what you see here is a dark field showing that whatever is bound to the plasma membrane got washed out. So that created the question, how would that be able to induce the cell death? So one of the targets we are focusing on is the Fas receptor. So we are suspecting that phycocyanin may be able to bind to the Fas receptor and activate what they call extrinsic pathway. And as you can see, that the combination of topotecan and phycocyanin was able to induce cell death, which was blocked by the Fas inhibitor. When we were using Fas inhibitor, for 3 hours, and then when we did the combination treatment, that inhibition was able to stop the effect, the extrinsic pathway activation by this combination, primarily by phycocyanin, was able to be blocked completely. So what is the role of topotecan? And the topotecan may be also able to induce extrinsic pathway to lead to cell death, and that was kind of blocked by the Fas inhibitor. So you can also see that the cleavage of PARP was inhibited by in the combination treatment by the Fas inhibitor. So this kind of indicated that it was not necessary for the whole phycocyanin to enter into the cells. Maybe by acting on the receptors, cell surface receptors, such as Fas receptor, it might be able to induce the extrinsic pathway through activation of caspase 8. It may be able to induce cell death, and that is what is shown in this diagram. So by activating the Fas receptor, it may be activating the caspases, and then it may be leading to activation of caspase 9 and 3, and leading to cytochrome C release, and which will eventually lead to cell death. And this is what we have confirmed when we treated xenograft animals, because once we completed the in vitro study, we wanted to see whether we are seeing any adverse drug effects in this during these combination treatments. And also we wanted to see whether this combination is working better than the monotherapies. And as you can see, phycocyanin, which was given in the drinking water to these xenograft animals, basically the A459 cell line was xenografted into these athymic mice, and we allowed for the tumor to develop for about a week. And after that, we treated them with phycocyanin in the drinking water, and whether it was Taxol or topotecan, it was injected intraperitoneally. And as you can see, that these drugs were able to really reduce the tumor burden. Phycocyanin was able to show some effect by itself. Phycocyanin was able to reduce the tumor burden, but when you let the animals to live longer than 60 days, in this group, phycocyanin group, the tumor started to grow more aggressively, and then eventually the animals died. In other two cases, that was not the case. That showed that phycocyanin can delay the growth of the cancer, not necessarily making the cancer to regress. So in terms of body weight, there were no significant changes in the body weight, which showed that there was some decrease in the Taxol, slight decreases in the Taxol and topotecan treatment up to 75 days. But with the phycocyanin, there was no significant changes in the body weight. And this is a picture showing the effects of the combinations. Here we showed how the combination with Taxol, phycocyanin, Taxol combination worked compared to the control. You can see that the tumor was growing more aggressively without any treatment. Phycocyanin was able to delay the growth. But when you combine that with Taxol or topotecan, we were able to see much significant therapeutic benefit. So this is kind of represented graphically in this picture. So basically what I'm trying to say is a lot of physicians are very hesitant to allow patients to take phytochemicals. Some of them, if they don't know about the benefits, they advise the patients not to take, in this case, for example, not to take any dietary supplements, because they may not know what kind of side effects it may produce. In order to take a safer approach, they recommend, tell us what you're taking. We will do some literature evaluation and then advise you whether you can continue to take it or not. But if we can do systematic studies, as we have done with ethanolic extract and with phycocyanin, we might be able to show to the scientific community that some of these phytochemicals can really work well with chemotherapeutic agents and provide the therapeutic benefits. So that was the original goal, and I think we have made significant contribution in showing to the world that this can be done. So let me go to the last 3 slides and introduce the rest of the team members here. So, we were able to get the funding from state of Florida for doing the lung cancer study, and Royal Dames of Cancer Research is one of our supporters. They are supporting our drug discovery research and other types of research that is ongoing at the Ramba Goodwin Institute for Cancer Research. And we have recently received funding from the Community Foundation of Harvard for looking at the effects of anti-cancer drugs on peripheral nervous system for inducing peripheral neuropathy, because this is one of the common occurrences in multiple myeloma patients. When peripheral neuropathy develops, they have to stop the treatments and put the patients on drug holidays, which makes the cancer to progress very rapidly. And when we come up with the treatment measurements, such as treatment measures, such as use of phytochemicals or other combinations, we might be able to continue the treatment without putting the patient on hold. So this is one of our programs. So now we are training a lot of students, undergraduate medical students, and those who are sitting in the front row are undergraduate students, whether they are coming from US or from outside. We would be able to take them and train them during the summertime. Unfortunately, due to COVID situation, we were not able to accept any students at this time. And this is our team. All of these are graduate students on the left side, and Dr. Thiagu, who might be online today listening to the presentation, and Dr. Minard, he is an expert in doing high throughput analysis. Dr. Uma, who's listening to the presentation, she's an expert in doing cell biology analysis, and these are the 3 graduate students who are with our team. And this is our vice president for operations, and he was visiting our institute recently, and we had a picture with him. And this is our team with all the members of the RGI. So I was going to show this video, but it may not work. And let me try, and looks like it is not going to work. So because of that, I'm going to go to the last slide and say thank you for your patience and listening to my presentation. If you have any questions, feel free to ask now. And I believe the moderators are going to receive the questions also and shoot the questions to me. Once again, thank you for your patience. Dr. Suresh, I'm not able to hear you because you are muted. There's a question in the chat. Yes.
Can you hear me?
Yes.
First of all, I'd like to congratulate Dr. Apu Ratnavelu for a wonderful talk. I think it was very informative, and I think the students can build on the fundamental knowledge that was taught to them by the teachers about the extrinsic and intrinsic pathways associated with cell death, and they can extend that to ethnopharmacology-based studies for drug development. Before I ask a few questions from my side, I thought I will give preference to the questions posed by the participants. First question is, does a particular phytochemical or combination, does a particular phytochemical or combination of 2 or more work for all types of cancer? Question posed by Shreya Churungu.
Dr. Shreya, good question. I wish I can say that there is a miracle phytochemical that is available that can cure all kinds of cancers. Not even chemotherapeutic agents that are currently available can cure all kinds of cancers. So we have to really see for which cancer this particular phytochemical is the most effective, and try to use for that kind of cancer. And as you can see, phycocyanin worked well for both prostate and lung cancer. It may not work for other types of cancer, such as ovarian cancer or liver cancer, pancreatic cancer. So it would be, somebody was asking about customizing the medication. It would be a good idea to see for which cancer these phytochemicals are going to be the most effective and target those cancers.
Thank you, sir. The next question is, what cell would you use to study the effect of these phytochemicals in normal cells, and what will be those effects? Question posed by Bhagya Lakshmi.
Bhagya Lakshmi, very nice question. We did use the normal epithelial cells I did not show the data. I believe it was in the presentation because the reviewers were very careful in asking what effects can you expect from phycocyanin on the normal cells, normal tissues. So, we did use the epithelial cells for testing the effects of phycocyanin. It was not really producing any significant side effects or toxicity. Very minimal cell death. Whatever cell death we observed were similar to what we typically see when the cells are growing without any treatment. So it's very safe. And the people, they, since this is a fluorescent, powder, they put it in drinks. And when you hit it under UV light, the drink fluoresces, and they love to drink those fluorescent drinks. That is how they consume in some of the parts of the world. The reason I'm mentioning about this is it's very safe. And when it is combined with Topotecan and Taxol, it was really potentiating the therapeutic benefits.
Okay, the next question is from Janatul Firdaus. Is caspase-3 gene expression the only way to detect apoptosis? If it is not expressed, does it mean apoptosis did not take place when cell viability showed 60% death in the MTT assay?
Janatul, nice question. Caspase measurement is one of the methods for detecting apoptosis. It's a robust method. It's easy to do, but I would not rely on MTT because Uma, who's listening to this presentation, she was able to really look at cell death through 3 different mechanisms. One is apoptosis, second one is necroptosis, and she was able to really detect the markers for necroptosis and also necrosis. All these 3 can show decrease in the cell number if you're doing by MTT assay. Whether it is 60% or more, MTT assay is not going to be able to differentiate. If you're going to be looking for apoptosis, caspase activation is one of the measurements. We also use PARP cleavage. Not necessarily BCL2 depletion can be seen in all apoptotic mechanisms. Sometimes you may see Bax elevation without decrease in BCL2 levels. And because of that, you can do caspase activity measurements, you can do PARP cleavage, and this is what we typically do. And finally, DNA fragmentation to confirm. And if you have a good confocal microscope, you can see the fragmentation of the nucleus also. That is what Uma is going to be doing in the next few weeks. So these are some of the measures to make your reviewers excited when they are looking at your papers.
There's a question from a faculty, SBST faculty, Dr. Kavita Tirumurugan. What phase of the cell cycle is affected by phycocyanin?
Good question, Dr. Tirumurugan. Good question. We are thinking that it may be blocking the transition of the G1/S phase, but there's no confirmation yet, and we are going to be determining which phase is exactly working. But if you look at topotecan really blocks the G2 phase. That is a well-known fact. Some of the topoisomerase inhibitors, they block S phase also, but we believe that phycocyanin may be, kind of blocking the transition from G1 to S phase, but no, we haven't confirmed that yet. Very good question.
Thank you, Dr. Apu Ratnavelu. Now, question about glioblastoma from Jill.
Yes.
Have you performed any studies regarding ability of natural compounds to cross the blood-brain barrier? Thank you.
I'm waiting for somebody to give me a natural compound so that we can test it. So we have a very unique system. And the system we use is xenograft model, where we implant tumor into the brain, and then once we close it, it grows in about a week. It grows to a measurable size, because we cannot let the tumor to grow to 200 milligram, 300 milligrams. It'll cause a lot of cranial pressure. It'll cause a lot of distress to the animals. So we have the imaging system which can detect when the tumor is established, measurable. So, and then we start the treatment. So this way, when we do the treatment, the drug has to go to the brain. It is not an intracranial injection. It is the IP injection or oral delivery, and the drug has to go to the brain and then stop the growth. And that is the system we have, which works very well. Actually, Mayo Clinic trained our scientist, Dr. Thiagu, who may be listening to this presentation. He went to Mayo Clinic and learned the system, and that is what we have. If anybody wants to collaborate, tell me this is the compound we would like to test, and I would be more than happy to collaborate as long as there is some kind of funding available from somewhere. But so far, we have not tested the natural products because this xenograft system is very expensive, and without funding, without good goals, we don't want to really spend our resources.
A couple of questions one from Sheetal Rana. How effective would be personalized? How effective personalized medicines are for the treatment of cancer? Sheetal Rana.
Sheetal, I don't know whether you're a doctor or Dr. Rana or Sheetal. Good question. Personalized medicine is the way to go. And I will give you one example, very quick example. Nowadays, as soon as they take out biopsy samples, they send it for genetic testing in the US. And they wait for the genetic testing to come out. Most of the profiling done nowadays are for cytochrome P450 system to make sure that the drug is not metabolized. And for example, if they are treating leukemia patients, they are looking for MGMT mutation polymorphism. MGMT polymorphism will determine whether the patient is going to tolerate the treatment or not. So basically nowadays, the genetic testing is done for determining whether the patient is going to tolerate the treatment. If the patient is going to be tolerating high dose treatment, then they would be getting the treatments, the benefits. So that's one of the approaches. If they are not going to be tolerating the drug treatment because the enzyme is mutated or it's mutated means it's not going to be metabolizing the drug, it's going to be causing side effects, right? So in addition to that, they are looking for target mutations also. If a particular target is present, is it mutated? Is it going to be showing response for this drug? So that is the way to go. And I hope this is done in India in some of the hospitals, but the future of medicine is depending on how we can customize the treatment based on the genetic makeup. And because of that, I support that very much.
Dr. Apu, I just want to ask one question from my side.
Yes,
you're working on this Tulsi extract and topotecan combination, and then you went on to Phycocyanin and the other combinations. What would be the next step in terms of improving the bioavailability?
Well, Dr. Suresh, we stopped studies with ethanolic extract and also with Phycocyanin because of lack of funding. Getting funding from NIH for testing natural products is not very, very easy because there is, tons of literature available, and they feel that unless something that is very unique, they may not be able to, support the studies. We feel that our translational approach is very, very beneficial because if you dig into the literature, you may not see this kind of combination systematically looking at induction of apoptosis and other mechanisms that are related to cell death. But getting funding is the most difficult thing. And if the funding is available, we can certainly look at the bioavailability and other aspects. For example, in a Phycocyanin, cyanobilin molecule can come out and it can go into the cells. And we haven't been able to do that study because of lack of funding. Right now, we are focusing on glioblastoma work because it's going to be attracting funding. We are focusing on peripheral neuropathy projects because we are going to be testing patients with that project, and that has given funding, and it's going to bring more funding in the future. So whatever is going to be benefiting the patients, and that is what NIH is going to be supporting. And if we can do the testing in India, it doesn't matter, whether it is bioavailability study or some other study with natural products, we can show that, and then we can attract funding.
Thank you, Dr. Ratnavelu. Before we end, one general question not related to cancer from Nirmal follows. Sir, could a natural product with immune boosting capability be helpful in treating coronavirus disease?
Actually, good question. Who was the
Nirmal,
general question?
Nirmal follows.
Nirmal Paulos, a very nice question, Nirmal. Actually, we submitted 2 proposals. Uma and Tiagu, they are co-investigators on those proposals, for treating COVID-19, one by using immunotherapeutic approach. What we are going to be doing is we are going to be creating a recombinant protein that will bind and neutralize COVID-19. And since this is going to resemble immunoglobulin, we are hoping that it can boost the immune system also. Now, of course, boosting the immune system is the best way to stop the multiplication of the virus. Even if the virus enters into the human body, which is becoming inevitable. If you look at the number of cases, not only in India and also in the US, one way or the other, virus is going to get into the body. And, our work should start from that point. We should be able to stop either by boosting the immune system or by developing therapeutics, which will be stopping the multiplication of the virus. From that perspective, I would say boosting the immune system is the best way. And this is the natural, process. And how we are going to be boosting the immune system? Are we going to be using any colony stimulating factors or some other therapeutic approaches? I don't know. This is what doctors have to test and come up with. But your point is very well taken, boosting the immune system. Are we going to be using natural products for boosting the immune system? At this point of time, I cannot think of any natural product that can boost the immune system to the level, the nilemembicudinib that was presented by one of the speakers, one of the junior faculty members, seemed to be used in many parts of the world. It seemed to be working because of the combination. So that is the approach, use of combination approach, combination of natural products may boost the immune system and help.
Dr. Ratnavelu, can you take one more question, sir?
No problem, sir. I start my presentation well in advance for the questions, so I can take one more.
Okay, thank you, Professor. Aritra Ganguly, will FACS be effective for detecting cancer? What kind of mice is used? Is Swiss albino mice applicable for implantation of phycocyanin? What type of cancer grows in these mice? Can we observe asides in the mice?
Aritra, good questions. FACS can be effective for detecting cancer if you're going to be looking for a particular target. So it can be a gene that is mutated, and particularly if there is a mutant protein that you are going to be tagging, or if there is a way of detecting that protein by FACS. Basically by FACS, you're going to be isolating the cells, and you can do the second level of confirmation once those cells are sorted. So that is the approach. And actually, many of the clinics, we're going to have a flow cytometer for the purpose of doing cancer detection, but you need to have some kind of antibody or some kind of marking of the cancer-inducing protein or gene. So that is the way it is going to help. In terms of using mice, and Swiss albino mice can be used if you can suppress the immune system. Because if you're putting the xenografts are basically human tumors, or the tumors developed from the human cancer cell lines. When you put these xenografts into mice, most likely they're going to be rejected if the immune system is not suppressed. So it's necessary to use immunocompromised mice, or you have to suppress the immune system, then you can put any kind of tumor in these animals. But when you are suppressing the immune system using chemicals, that chemical may interfere with your study when you're looking at the anti-cancer properties. So you have to be very careful. There are a lot of immunocompromised mice available. in India also, you should be able to use that for your studies. Did I answer your question?
Yeah, I think so. I think so. Thank you. Thank you. On behalf of the organizers, I'd like to thank Dr. Apu Ratnavelu for a wonderful lecture. I think the students would have also been benefited from the results and also the experimental design. And some of the students would have been able to relate to the extrinsic and intrinsic pathways of cell death that are taught to them by their teachers. Thank you, Dr. Apu Ratnavelu. And I want to thank the organizers for having given me this opportunity to chair this session and listen to the wonderful lecture from Dr. Apu Ratnavelu. Over to Professor Jabez.
Professor Suresh, thank you very much, sir. Thank you for your patience and thank you for handling the session so well. And I also thank Dr. Apu Ratnavelu for his session. Thank you very much, sir, once more. So now we'll be moving on to the next session, which is the poster evaluation. So it starts right now from 5:30 onwards. So those who are presenting the posters can directly go and join the link and start your presentations. And if anybody else is having any other queries, I'll be here for another couple of minutes. So you can just post your question in the chat box so that I'll be there to help you out for just another 2 more minutes. Thank you very much. And tomorrow's session starts exactly at 10 a.m. and the first presenter speaker is Dr. Challa Suresh. So I request all of you to join by 9:45 itself sharp, so that the entire session will go on time. Thank you very much. Thank you all the participants. If you do not have any more questions, you're free to leave the day's talk session. Thank you.
Thank you.